Greetings, ladies and gentlemen. Today I would like to discuss two major recent advances made in Minnesota and France that will help human beings combat the ravages of aging, or senescence, in both animals and humans. These advances, like many scientific discoveries, are just first steps, but eventually we would hope that they will result in widely available, highly effective treatments for repairing damage and extending both chronological and healthy lifespans in humans. The first of these studies was made in the Mayo Clinic in Minnesota. The authors were Darren J. Baker, not to be confused with David Baker of the University of Washington, who is responsible for FOLDIT. Darren J. Baker is a different researcher, and Jan M. van Deursen, who led a team of researchers to work on a strain of mice engineered to have progeria. Now, progeria is a very unfortunate condition. A small percentage of humans have it as well. It's a genetic disease that results in faster than normal senescence. So essentially, the lifespans of organisms with progeria are considerably shorter than the lifespans of corresponding organisms without it, and furthermore, the symptoms of senescence are manifest at much earlier ages in organisms with progeria. The researchers targeted specifically the senescent cells in these mice, and they identified a particular molecule, p16-inc4a, that the senescent cells produce, but the healthy cells do not produce. The mice were also genetically engineered in another way, with a second gene that produced a protein that would specifically kill the cells producing this molecule, p16-inc4a. And this protein only targeted the senescent cells and left the younger cells alone. The younger cells had not divided as many times, and they did not produce this molecule. Also, they did not produce other harmful byproducts of senescence. So only the younger cells were left in these tissues. So what were the results of this treatment? The mice had overall healthier organisms. Their muscles remained effective for much longer. They lost less fatty tissue with age, so generally their tissues remained more robust. The mice that did not initially have cataracts did not develop those cataracts even as they became older, and even older mice that began to receive this treatment late in their lives were able to remain youthful longer, at least to retain youthful characteristics in many of their tissues. So their tissues didn't degenerate as much. If they already had cataracts or other problems, this treatment didn't reverse those problems, but it did stall further decay. Now, a big question for this study was it able to actually increase the lifespans of these experimental mice? The answer is no, but the reason why the answer is no is because of the very design of this experiment that precluded the extension of such lifespans. And really, these mice were designed to die at early ages. It's almost deliberate because the strain is short-lived. You can perform experiments on it within a shorter time frame and get your conclusions faster. The removal of the senescent cells also did not stop the deterioration of the blood vessels and the heart tissue in these mice, and heart failure is what these mice typically perish from in the first place. So because this particular treatment did not address heart failure, the mice died at roughly the same age as they would have without the treatment. However, while they were alive, they were significantly healthier than they otherwise would have been. So their lifespans remained short, but they were of a higher quality than they would have been otherwise. The Mayo Clinic explicitly plans to perform this experiment again, this time with normal-lived mice of a lifespan of 3 years or greater. And this is where the conclusions will really be quite fascinating, because if this treatment is also able to extend not just healthy lifespan, but chronological lifespan, it will be absolutely groundbreaking. As it is, this is still quite a major advance. It shows that senescent cells damage the cells around them. It's not just the individual cell that becomes less functional and atrophies. Also, it has negative external effects on other cells. This is a hypothesis that existed in the biological community before, but had not been explicitly proven up until now. And so removal of these senescent cells can greatly delay the deterioration of many tissues, though apparently, as this experiment suggests, not all of them, and it can increase the span of healthy life. If similar treatments are developed in the future for humans, it would be very beneficial, even if chronological lifespans aren't expanded considerably, though we would of course hope that they will be by this or other means. Still, to have elements of youthful vitality for many more years or even decades would be absolutely amazing and would certainly transform human lives and entire societies for the better. The second study that I would like to focus on was performed in France by Jean-Marc Lemaitre of the French Institute of Functional Genomics. In this experiment, human cells in particular were the subject. Cells were collected from people very advanced in terms of senescence, aged 74 to 101 years. And these senescent cells were successfully reprogrammed into induced pluripotent stem cells, or iPSCs. These induced pluripotent stem cells have the same characteristics as embryonic stem cells, and that includes their versatility. In 2007, for the first time, it was discovered that it is possible to create pluripotent stem cells from adult cells. You didn't have to have embryonic stem cells in order to create cells that can then be transformed into more differentiated cells within the human body. This is very significant because it provides an avenue for creating stem cells that doesn't require the destruction of embryos, and in the future could be much easier than harvesting stem cells from embryos. So in this study, 6 transcription factors were used to create the pluripotent stem cells. The transcription factors are proteins. They bind to specific sequences of DNA, and they regulate the transfer of information from DNA to messenger RNA. And the 6 proteins are OCT4, SOX2, CMYC, KLF4, NanoG, and Lin28. The stem cells can produce differentiated cells for a wide array of specific human functions. This is very significant. In theory, one could turn a stem cell into the cell of a heart, or the cell of a lung, or the cell of a liver, or any other organ that might fail or require rejuvenation in the future. So the potential is quite vast. And what's particularly significant is these stem cells do not reflect the history that they had. They do not reflect their past as senescent cells. This is extremely important for facilitating genetic engineering and rejuvenation of older individuals in such a way that their immune systems will not reject the new cells, because these cells will have the exact same genetic material as the originals, but because they were created from stem cells that are identical in terms of their potential to embryonic stem cells, it will be possible to give these older individuals young cells that haven't been affected by the ravages of senescence. So what are some of the implications of these new discoveries? First of all, they're major advances in understanding. How does senescence inflict its damage? And furthermore, how can we stall some of that decay, and how can we reverse it? Of course, therapies have not yet been developed for human beings to build off of these discoveries, and it will take years to develop these therapies. However, in the future, they could be targeted toward destroying just the senescent cells in humans and leaving the healthy ones alone. Do as little collateral damage as possible, and therefore maintain vitality without adverse side effects. Furthermore, The Lemaitre study could enable humans of any age to have stem cells created from any cell in their body, and then be used to develop healthy tissues to replace ailing ones. And all of this could be done without the threat of the immune system rejecting the new tissues. These new tissues could be created even to replace the tissues that would not be helped by the removal of senescent cells. So for instance, if the Mayo Clinic study's conclusion that the heart and blood vessel tissues were not affected by their treatment holds in subsequent treatments and in human trials, this would still not be an insurmountable barrier because the new tissues could be created from pluripotent stem cells to replace senescent heart and blood vessel tissues. To summarize, the significance of this is people could senesce much slower than they currently do, and once they do senesce, they could get fully compatible replacement tissues. The combination of these two factors would mean dramatic extension of human lifespans. Once again, this is not yet a present medical reality. However, one can hope that these advances will motivate further research into these areas, development of further treatments, and a rush to commercialize these treatments at the earliest feasible date once it's ascertained that they can be performed safely and cost-effectively. When this happens, significant barriers to human longevity and quality of life could be lifted, and that could be truly revolutionary. I encourage you to explore the articles that I've linked to in this section, and there are many more on these particular studies. This is just a sampling, but it will provide further details to those who are interested and will also hopefully motivate some of you to explore these issues further. Thank you very much.